Okay, friends, so this is the first episode in our next unit. This next unit is on DNA mitosis and protein synthesis. And so really what we're going to focus in here, before we talk about DNA and what DNA does, we're going to talk about the cell cycle and give you a little bit of context of what's happening inside the cell and why. So really we're going to talk about these two parts of the cell cycle in a little more detail: interphase and mitosis. So these should be familiar terms from middle school. If they're not, then you need to make sure that you come to me with questions of things that you don't understand, because I'm just going to be reviewing things and adding some extra detail to what you were expected to know to pass last year's SOL. So we're going to talk about interphase and mitosis. So this is what you need to be able to do. You can describe the cell cycle, and can describe and recognize the 4 stages of mitosis. So let's go and get started talking about the cell cycle. So the cell cycle, well, it's a cycle, so I'm going to go ahead and draw as best of a circle. It looks more like an egg, but that's okay. You can draw a better circle than that, I hope. So go and draw your circle. I'm drawing arrows that are going to go around this circle clockwise, so you know what direction things move in our cycle here. And what you should know is that a very small section of the cell cycle is mitosis. We're going to talk a little bit about what mitosis is in a few minutes, but a very small section of it is mitosis. The other section, the rest of it, is interphase. Sorry, I'm writing this sideways. So it just wraps around. So most of the cell's life actually occurs during interphase, and actually interphase is broken up into 3 separate sections known as G1, S, and G2. And G doesn't stand for growth, but it is one way to think about it. So let's say that we, in G1, I'll tell you what happens in G1. In G1, the organelles, remember from 2 units ago, all those organelles that we learned about? They double. So they increase in size, they split in half, so we have twice as many organelles like mitochondria. If this is a plant cell, we're talking about extra chloroplasts, extra endoplasmic reticula, Golgi bodies, that kind of thing. In the S phase, DNA replication occurs. And we're going to talk a little bit about that soon, but we're going to hold off on that discussion now. So I'm going to put a little star here, so we know that we're going to come back to this idea. But DNA replication occurs here. Sometimes it's called DNA synthesis. But basically DNA just gets copied here, and in G2 the cell checks itself, meaning it asks itself 2 questions, and this is only if we think of cells as conscious things. So I'm giving them this ability to ask questions. Obviously cells individually can't do that. So the cell checks itself. So it asks itself, did I copy the DNA correctly? So in other words, it's proofreading, which we don't do so well, right? But anyways, did I copy DNA correctly, and do I have enough organelles to split into two smaller cells. And if the answers to these two questions are yes, then the cell will continue around the cycle into mitosis. And that's what we're getting ready to talk about. So this is the cell cycle. I didn't label that for you. So this is the cell cycle, and what's happening here is, what this diagram is showing is a relative amount of time that a cell spends in interphase versus mitosis, and what happens in each part of interphase. So we would expect more DNA in G2 than in G1, because between G1 and G2, S has occurred, and the cell in G2 is going to be bigger because during this process of interphase, the cell has been growing because the organelles are doubling. So there needs to be more room for those organelles to live in, I guess, quote unquote live, before the cell gets ready to go through mitosis. So let's talk about mitosis in a little bit more detail. Oops, excuse me. Here we are. Okay. Mitosis. And really, you should know that, well, let's say this. You might think that you know what mitosis is, but you can't learn anything that you think you already know. So let's make this very clear what mitosis is not. It is not equal to cell division. Mitosis is not about cellular division. What mitosis is nuclear division. This is division of the nucleus. And so if we're talking about the nucleus, let's make a note that this is really only for eukaryotic cells. Now I hope you're realizing that all this material is building on itself from previous units. So if you don't remember it, you do need to go back and look at it, because in order to be successful in this class, you need to master all of the material. Just because we're not talking about it anymore in great detail doesn't mean you're not expected to know it. So nuclear division is what happens in mitosis. So let me draw what happens in mitosis. So the first phase of mitosis, draw my typical cell here, and my nuclear membrane, but it's going to be dotted. Actually, let me go back. I just wanted to erase that so that I could draw them on a new page to make it a little bit bigger for you. So let me draw a cell here with our nuclear membrane. And remember that there's going to be a lot of other stuff inside that cell that I'm not drawing. This cell, as a eukaryote, will have mitochondria, it will have ribosomes, an endoplasm in particular, rough and smooth, Golgi bodies, vesicles, the whole shebang. It'll have a lot of stuff, but really all we're concerned about with mitosis is the nucleus, okay? So if we've got our nucleus here, I'm going to draw in several different chromosomes here. So I hope you remember the shape of the chromosomes from middle school, maybe elementary school. And what I'm doing is drawing 4 of them, but 2 of them are bigger than the other 2. And that's okay, that'll come into play a little later, but make sure you draw it that way, and you might want to indicate their color in your drawings with a color pencil marker, highlighter, whatever. So you have 2 separate colors, but what I want you to do is jot down a little bit about what I described about what's happening here, because I'm not going to write it all down. So we've got our first cell here, and this is the phase called prophase. So the first part of mitosis, the nuclear membrane starts to disappear, it starts to break apart. Remember, the nuclear membrane is made out of phospholipids, so those break apart. The chromosomes start to become visible because they have condensed down. What that means is DNA, which is really loose, usually during interphase, starts to wrap itself around itself and other proteins. So it's kind of like string being wrapped around to become yarn, and becomes thicker, so that then we can see it actually under a microscope looking like this. So this is what happens in prophase, but we also have another component. We have these things called centrioles. I hope you remember that word from the cell organelle unit, so 2 units back. They appear and they double, and we're going to see them reappear in the next phase. The cell is going to be a slightly different shape. In the second phase, which is called metaphase, the centrioles have moved to opposite ends of the cell, and they've created, well, let me draw my chromosomes here first. Here's my big chromosome, there's my little red one, here's my big blue one, and my little blue one. Now, these are not actually colored under the microscope, but this is going to help us keep track of them when we talk about another process in the future. So, oh, excuse me. Sorry about that. That was my watch going off as an alarm. So these chromosomes are not actually colored underneath them in the cell, but they're just going to help us keep track of something in the future with another process. What we see with these centrioles, what they're doing is actually creating spindles across, these basically just fibers that go across the cell, attaching to these chromosomes. So they're lined up here in the middle. And right here in the middle, they're lined up. I'm just drawing another dotted line. This line doesn't actually exist, the white one. And that white line is what we call the metaphase plate. So it's just all these chromosomes are lining right up in the middle. And then in the next phase, just drawing a little longer to give myself some more space, we've still got our centrioles on the sides. And what we see is those chromosomes got pulled apart from each other. So the X actually got pulled apart because the spindles that went across the cell, oops, I forgot to draw that chromosome. I'll come back and add that. Actually, I feel a little bit like Bob Ross right now. If you don't know who that is, look him up. But these chromosomes have gotten pulled apart in this third phase that we call anaphase. And we'll come back to this in just a few minutes. We'll come back to this in just a few minutes. So, those chromosomes have gotten pulled apart, and so what we see in the last phase of mitosis is actually 2 nuclear membranes that are forming around these 4, they kind of look like half chromosomes, but there's something that's very important about them that makes them their own chromosome. But This part is called telophase. And then the last, so these are the 4 parts of mitosis, and here's a good way to remember them. PMAP, prophase, metaphase, anaphase, and telophase. So prophase, metaphase, anaphase, telophase are the 4 phases of mitosis, but this is not the end of the process. So remember that mitosis is nuclear division. Our nucleus starts to disappear in prophase, centrioles start to move to the edges, chromosomes start to condense on themselves. In metaphase, by the time we've hit metaphase, the chromosomes have lined up on the metaphase plate, the centrioles are on opposite sides of the cell, and spindles have formed across the cells, attaching to the chromosomes, and by anaphase, those chromosomes have been pulled apart from each other. Now they're pulled apart to the opposite ends of the cell, and by telophase, the nuclear membrane starts to reappear around the separated chromosomes. We have 2 new nuclei by the end of this process from the one. Now there is a key difference that there's only half of that X, but we'll come back to that in just a minute. The other part that happens just after mitosis, so I'm going to draw 2 lines just so you know this is a separate process, and it's called cytokinesis. I'm going to draw it as best as I can. Alright, so cyto, you know, like endocytosis, exocytosis, cytoplasm refers to cell. And kinesis is like kinetic energy. It means moving or movement. So the cells are moving, they're actually moving apart from each other. So what we end up seeing inside, we've got our nucleus, nuclei rather, right? And they actually have started to, the cells have started to pull apart from each other, which is quite interesting. And this is the part of cellular division. And again, this is a separate process. This is not mitosis. This is cytokinesis. This is how cells separate. So sometimes we can have nuclear division or mitosis without cytokinesis. And that would result in multiple nuclei inside of one cell. And that happens from time to time. It's quite interesting when it happens, but most of the time we have mitosis that's immediately followed by cytokinesis, which is the cell division. Okay? Now, let's go back and talk about what's actually happening here in anaphase, and we have to talk about one other little detail about what this chromosome is. So what we're going to talk about very quickly as a side note is the anatomy of a chromosome. Now, we drew the chromosome, I'm just going to use this red color, like an X. And what you should know are a couple key parts to it. Know that, in case you don't already, maybe you don't know this, is that a chromosome is made out of DNA. That's one of the vocab words, so you should know what that is and what it stands for. But it's made out of DNA plus proteins around which the DNA has wrapped itself around. So it's become like yarn, it's really thick, and then these 2 different sides of the X are called sister chromatids. Those sister chromatids are copies of DNA. So that means this side on the left is an exact copy of the one on the right. Unless a mutation occurred, but the chances of that are pretty low because the cell has done its proofreading before mitosis. Remember, G2 does the proofreading, although sometimes mistakes happen. For example, if you're writing down notes from what I'm writing, there is the potential that you make a mistake in what you write or what you copy. even though you're trying to copy things word for word or write down things that I say word for word, you might still make mistakes. And same thing happens with DNA when a copy is made. Now, right in the middle of the chromosome is another part that we call a centromere. And this is where spindles attach. So where spindles attach. So remember that in metaphase and in anaphase, in metaphase the chromosomes lined up on the metaphase plate using the spindles that were created from the centrioles on either side of the cell. And then these sister chromatids are actually what get pulled apart from each other in anaphase. And they get pulled apart in anaphase because the spindles have attached to the centromere and pulled the two sister chromatids in separate directions. But remember that sister chromatids are actually copies of each other. So when they get pulled apart, so when these two parts get pulled apart, so the X is apart, these are actually, because they're both copies of the chromosome, we actually end up with two exact copies of the nucleus. So let me go back to our diagram here. Here, after the chromosome has been, the sister chromatids have been pulled apart in anaphase, these two resulting nuclei, these here, this complete nucleus and this one, actually have the exact same DNA, unless a mutation occurred, because the sister chromatids were what separated, which are, again, are exact copies of DNA. Now notice, though, in cytokinesis, that we only have one half of the X chromosome. Well, if you can imagine that this cell will continue to split down the middle, this cell will continue to split down the middle so that a membrane would form here, separating the two cells, and then the cell starts the cell cycle all over again. So let's just follow this one on the left. So that cell is going to go through the cell cycle. Again, it just finished mitosis. So let's add, we've got our 4 stages, right? Prophase, metaphase, anaphase, telophase, PMAT. It just finished that, so we've got 2 separate cells. Sorry, we've got one more little thing to add. It's not part of interphase. If you remember, this is cytokinesis. So after cytokinesis, we result with 2 separate cells. Both of these cells are going to continue in the cell cycle. If we're following the one on the left, it's going to go on its own through G1, in which the organelles develop and double. Then S phase, which is where DNA replication occurs. Interestingly, this is where the sister chromatid was made. That's where the exact copy, that second half of the X, came from. And then it goes through G2, where it asks the questions: did I copy the DNA correctly? Do I have enough organelles to split into 2 separate cells? And again, if there are check marks to that, yep, it continues, and then that cell divides again through prophase, metaphase, anaphase, telophase, or mitosis, and then cytokinesis, making 2 new cells. So in mitosis, what's really interesting is that 2 exact copies of the nucleus are made. 2 exact copies because of what happened here in S phase. And we're going to talk a little more detail about this DNA replication and how that happens, so that every time that a nucleus divides in preparation for cell division or cytokinesis, we can get those sister chromatids to split apart. Okay, so I hope I gave you quite a bit of detail. Let me go back here and just double check I hit everything I wanted to hit on. Yep, so again, think about these chromosomes inside of cytokinesis, inside of the nucleus in cytokinesis, just about to go through G1, and then S where the extra sister chromatid is going to come from, and then it's going to go through G2, and then prophase, metaphase, anaphase, telophase. And think about this in a larger sense, that you actually started from one cell. A sperm and an egg came together to give you all the chromosomes. That one cell immediately started to go through the cell cycle. It started to go through G1, S, G2, then split into two cells. Then each of those two cells went through G1, S, G2, and mitosis, and split into two cells each. So now you have 4 cells. Those 4 cells are going to go through G1, S, G2, then mitosis, and cytokinesis, and those 4 cells become 8. And it continues and continues and continues until we have the trillions of cells that make you up today. And it's all because of mitosis. So if you've actually looked at some of your cells under a microscope, you would see some cells going through mitosis. And really the ones that we would see going through mitosis most often are the ones that are in which you are growing more cells quickly, or when you have to repair tissue if you get hurt. So like when you cut yourself, your body makes more skin cells to replace the skin cells that you lost or damaged. So those cells are going to go through mitosis to make those new cells to replace the ones that were lost or damaged. Okay? If you have any questions, jot them down. We'll talk about them as soon as I see you again. Okay? Let's double check that we can do these things. You can describe the cell cycle. I hope you can. We've gone into actually quite a bit of detail on that. And that you can describe and recognize the 4 stages of mitosis. A good way to practice this would be to draw and describe them on your own, and then practice with somebody. And make sure that you know these, or do flashcards of these phases and be able to talk about them and what's happening. That would be a good check for yourself to make sure you are ready and can move on to the next set of learning targets. Okay?